Hi, this is Deb, and I'm going to walk you through Chapter 5 of PowerPoint. I've modified the text version a little bit with some examples, as you'll see coming up. First off, what is energy? Energy is a capacity to do work. We'll have either potential, which is stored energy, or kinetic energy, which is energy of motion. So if something is moving, we're talking about kinetic. And the two forms go back and forth, as you could imagine. When you're climbing up a hill, kinetic energy, you're moving, using that energy. Potential energy, when you're at the top of the hill, it's only going to take a small activation to send you down. And then the movement going down is kinetic energy. Let's look at some examples. Your breakfast, potential or kinetic? That's potential. It's actually chemical potential. So the molecules store all kinds of energy in the bonds that you will later use. Walking across the room, kinetic. That's movement. So you ate your breakfast, stored that energy stored in your food, then you walk across the room and you use it. Diving off a diving board, kinetic. A ball poised at the top of the stairs. That's potential because it's only going to take a small activation to send it down those stairs. So just being at a higher elevation gives it potential energy. Cliff diver standing on the cliff, same thing, potential. A piece of pie, chemical potential because that energy is stored in the bonds of the molecules in that pie. The old-fashioned mousetrap that's set, when you bend that string over, spring down and hold it, that's a wonderful version of potential. You can see that it takes a tiny activation to set that off. Cheese as bait in the mousetrap, once again, potential, because we're talking about the energy stored in the cheese. And the mousetrap snapping and catching a mouse. We got kinetic going on. So how do we measure energy? We measure it in calories. And when you talk about, oh, my diet is 2,000 calories, like you did on your last, on the nutrition lab, we kind of use that number erroneously. A calorie is actually the amount of heat required to raise the temperature of 1 gram of water by 1 degree. And so now we have a kilocalorie. When we talk about calories in our food, we're really talking about kilocalories. Each one of those are 1,000. So it's a way that we kind of simplify it, I guess. So when we're talking about energy, we got some laws. We got some things that don't change. So we have the law of conservation of energy. That means energy can't be decreed or destroyed. You can't make it out of nothing. You can't destroy it, but you change it from one form to another. So say we put some gasoline on our car, turn our car on, the gasoline's burned to turn the pistons in the engine. We just transform that energy from gasoline potential to kinetic, moving those pistons. They cannot be changed from one form to another without a loss of usable energy. That's another law that doesn't change. So as we go from gasoline to turning the pistons, we lose a whole lot of energy as heat. And that heat is the loss that we talk about. And which one's more efficient? We are. Our cells are about 40% efficient, so we only lose about 60% as heat. Automobiles are much, much worse. They lose much more energy from those fuel bonds as heat. And heat, not usable. That's just released to the environment. Okay, one of my favorite words, entropy. That really means chaos. That means disorder. And that means our universe naturally lends towards disorganization. And to keep things organized, we have to input energy to keep it going on. So chaos will reign, most definitely, unless we put energy into it. So how do we do that? Energy is provided by the sun, and the plants make glucose, and that puts it in a more organized fashion from water and carbon dioxide. So it makes it more organized, and it takes energy to do that. Another simple example is the bedroom. Your bedroom stays nice only if you put energy into it to keep it nice. It easily goes into this phase without any effort at all. So how do we use energy? What is the currency for energy in our cells? Adenine triphosphate. That's the last form it takes for us to use it, and that's called ATP. That's one nucleotide with 3 phosphates, and I'll show you a picture in a second. It's a continuous cycle between ATP and ADP, adenine diphosphate. So the energy currency, the last step of where we use this energy, where we spend it, where we save it is ATP and ADP, that's cycle. And so ATP means adenine triphosphate, tri means 3, so here it is right here. We have adenine, the base, here's a sugar, ribose, and then we have 1, 2, 3 phosphates hooked on there. Turns out these first 2 phosphates are pretty stable. But to put this last one on, it's kind of like setting a mousetrap. It's barely hanging on there, and it takes a lot of energy to put it on. So as a result, it's darn easy to pop it off. And so that's what we do. Thousands and thousands of times a day in every one of our cells. We pop off that phosphate, and when we do that, the energy that was holding it together is released. And that's what we use. We're using it over here, sorry. So that's the energy that we use. We take that energy and we use it to walk, we use it to move, we use it to think, we use it to breathe, whatever we're doing. And then that is recycled. Energy from the sun is put into it, hooked back on. We need the energy, we pop it off. Energies from food. If you were a plant, you're doing it from the sun. Energy from food, hooks it back on. It's unstable, boom, pops off, stable. So ATP, adenine triphosphate, is very unstable. Adenine diphosphate, di means 2, so that means we've got 2 phosphates. That's very stable. That's not gonna do anything there. So that's more of this. It's just an easy coupled energy that is often with something else that we need to do for cellular work. So energy is released from what you had for breakfast. You break it down through respiration, put it in that last phosphate. Oop, you need it now to build some protein, to grow some hair, to grow some fingernails. And boom, we pop it off. Then we put it back on and we take it off. Like I said, thousands of times a day. And here's another example. And we couple it often in our bodies. An energy releasing reaction is hand in hand with one that needs energy, such as muscle movement. And it actually works at a very small level with myosin. It's almost a physical thing that makes that whole thing contract. It's pretty darn cool, actually, popping off that phosphate. All right, so energy. We've got a chloroplast and mitochondria being the main 2 organelles. We got photosynthesis going on, that plants take that energy and put it into a carbohydrate. They capture it. Cellular respiration, everybody does it, plants and animals, we break it down, and finally that energy is put into an ATP to be used. All right, so that's that big picture. So the flow of energy in the biosphere is going to be solar energy to a chloroplast and then to the mitochondria where it's broken down. So we've got solar energy coming here, and this guy turns it into a carbohydrate. And then this guy breaks it down and puts it into ATP, releases a bunch of heat. Remember, we're not that efficient. And the byproducts of that, carbon dioxide and water, go up here. And guess what? They're what's needed for this guy here to make oxygen and a carbohydrate. Pretty darn cool process, actually. How we manage all these, there's an enormous number of chemical reactions going on in our body at any time, and that whole thing of cycling molecules and energy is called our metabolism. So how are they managed? Metabolic pathways are managed by enzymes. Here we have one enzyme, enzyme 2, enzyme 3. Those are all different enzymes. That means to get this guy to turn into that, this enzyme's going to do it, and it's only specific for that. Then this guy is specific for making this happen. This guy is specific for making that happen. So it's a series of linked reactions that are very, very specific. They don't make a reaction happen, they just speed up the reactions. So they happen at a rate that will keep us alive, for example. And watch this on your own time. I'm not going to put it into here. So now we have enzymatic action. So what happens is we have our substrate goes into the active site of the enzyme, which fits it perfectly, like a lock and a key. And then when those guys are in there, this is one compound, and when they're in there, it kind of puts them in close proximity, so that they actually react like they're supposed to. And the 2 products come off. So I like to kind of think of it in a mechanical way. You have this substrate that either needs to be broken down into two products or perhaps combined to make one product. Building and breaking down both happen this way. And when it fits into these little sites, exactly, I think it puts all the players in close contact with each other so the reaction occurs. Enzymes are very specific, as you can see, a lock and a key, so they are named by the substrate they work on. Whatever that substrate is that they're working on, that's what their name is, with an ASE on the end usually. Such as this. So lipid, lipidase, urea, urease, maltose, maltase. Get the drift, yep. So I talked about it being a lock and a key. It's very specific to that one thing. It doesn't do anything but that. And what we're going to find out, this enzyme is a protein, so that three-dimensional shape for it to work is so important. If it's subjected to high heat, pH changes, the bond can change, it can come unraveled, and it no longer fits. No longer is a three-dimensional shape that works. That's why you can't, it's dangerous for you to have a high temperature, like 107 degrees, they'll pack you in ice, because they're afraid that the enzymes in your brain will start denaturing and then will no longer work, and that's when you die. So, all right. We also think it's an induced fit model. We see it actually. That as those substrate particles, molecules move into that, there's a slight shape change to accommodate it. So it kind of induces a new size and fits everything together. Important to note, enzymes are not used up by the reaction at all, zero. They can go on forever and ever and ever. And here's another picture of the induced fit. So we have our substrate coming in, going into the active site, then it becomes a substrate enzyme complex together in that new, slightly different shape. All right, watch that. And so we got to be able to inhibit sometimes. Sometimes we need to, sometimes it's a poison doing it, but we'll talk about how the two main things. There's a competitive inhibitor, something that fits the active site, but it's a different molecule. It goes in there, and so boom, the substrate can't go in anymore, and it's not going to work. A non-competitive inhibitor means this molecule fits in on a different side of the molecule, but by going in there, it changes the shape of the active site, so it no longer fits the substrate. So once again, it's not going to do its job anymore. Poisons are these, irreversible. Cyanide does that very thing. Penicillin does it to bacteria, not to us, but bacteria. It doesn't kill bacteria cells. It only stops them with the enzyme for building new ones with cell walls. So once you start taking penicillin, it won't kill what you got. Your body has to take care of that. But it won't let that bacteria continue to produce new little organisms because it cannot build a cell wall because the enzyme is not functioning. We have feedback inhibition. That's what we use all the time. When we don't have enough of something, we need to know that our body needs to make more of it. And so we have this kind of thing going on. So we have a substrate, boom, 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 end product comes up. When we have a whole bunch of our end product and we don't need any more, it fits into here and then changes the shape of the whole thing so it no longer works. So when you have a bunch of product built up, you don't need more product. And this is the feedback that tells us that. And this will talk about that too. So what do enzymes do? Energy of activation. It's like that little ball at the top of the hill. We need that little push to push it off the hill. Without an enzyme, it takes a whole bunch more energy for that to happen. With enzyme, things are closer, doesn't take so much energy, and it happens with less energy. That's why it speeds up a reaction. So, why can we only break down starch and not cellulose? Both are made of many glucose molecules strung together. Well, turns out our enzyme only works on this kind. If it's this kind of bond with one bond above the sugar ring and one below, because these are sugar rings here, then our enzyme doesn't fit that one. So we cannot break down cellulose. So good for you to eat cellulose. It's fiber in your diet. Definitely important for your digestive system. So cellulose serves a purpose, most definitely, in our bodies, and we need to eat it. That's the fiber in our diet, but we don't break it down to release energy like we do from a starch molecule, for example. All right, so we have to transport things in and out of our cells so that we can get nutrients and things we need into it and waste out of it. And so a plasma membrane acts as that regulator. It's selectively permeable. That means only some things can pass through freely. Some need transportation, and some are prohibited. So we have passive transport. No additional energy is required. We have active transport. Additional energy is required. And substances move naturally in passive transport from higher concentration to lower. But sometimes we need things to move from lower to higher, and that's when we have to add the energy, and that's where active transport fits into the picture. And sometimes we need some just kind of generalized transport going on. So let's talk about passive transport. Key point, no energy is required. There's no membrane required for simple diffusion. It just diffuses across there. Molecules move down their concentration gradient. That means they naturally go from concentrated level to spread out to everything's even, but they're moving to the lower concentration. That's the concentration gradient. What kind of things move fairly freely through there? Oxygen, carbon dioxide, and other small non-polar molecules. Water moves through because it has an aquapore, a special thing to go through. But of course, oxygen and carbon dioxide, that's pretty darn important, and what a gift that moves through freely. So the size, these guys might be larger. The concentration, if there's too many, if they're polar or not, all of those kinds of things can really impact if they're going to move across that shell easily. And if they are these kinds over here, we have special things that'll help us get it across. But first, let's talk about passive transport. We also can have facilitated diffusion. That means no energy is required, but it uses a transport protein to move it across. Water uses aquaporins, so it doesn't take energy, but it's got a special transport thing going on there. So we're talking about these. Here's our cell membrane image. 2 rows of those phospholipids, phosphates on the outside and inside. 2 layers of them. Then these lipids inside make them nonpolar. Nothing can go through there. And so we need these big giant proteins that either channel them through or carry them through. And these are going to be specific for a certain kind of molecule. Simple diffusion is going to happen everywhere, living, non-living, everything. That's just the whole process of something spreading out from concentrated to low concentrated. If I released a perfume at the front of a class, I opened the bottle, over time, the students across the classroom would smell it. We could see that going across the room because it's diffusing out. So it goes with the concentration gradient from high concentration to low. No energy is required. And you want to watch these. Let's see if I can get past it. You'll want to watch those. I went ahead and went through them quickly so that I didn't take up this time. There are several problems in our exam like this. I have a few coming up in this PowerPoint as well. But what we have here is a concentration, a 10% concentration of, let's say, sugar inside this tube inside here with a permeable, semi-permeable membrane across the bottom. And there are only 5% sugar solution in the beaker. So sugar content's higher in the tube, water percentage ratio is lower, and that's the opposite in the bottom. So what water is going to do, because water is going to move, it's going to go into that tube to try to balance out. So there's the same number of molecules of water, same ratio of water to solute. And so we have this water level going up because water is going to be doing that. And watch this one. And then we have the effect of osmosis on cells. This is kind of cool. As an animal, here's our red blood cells. We want isotonic, both the same. So the water goes in at the same rate as it comes out. So this cell is not gaining water, nor is it losing water. That's the concentration we shoot for in our world. So isotonic is what animals like for our cells. That's the perfect thing. Not so for plants. This is a plant on the bottom, and you'll see the cell wall, and it's rectangular, and the central vacuole inside here. It turns out that that central vacuole has to be super full for a plant to not be wilted. And so they like a hypotonic situation. So an isotonic situation with your plant here would look a little bit wilted. So hypotonic. I always like to think of it, I have to admit, I make simple little things to remember. Hypo, hippo, big and round. So if it's put in a solution where there's a greater concentration of water outside the cell, the water's going to go in that cell to balance it out. In animals, that's bad news. I think I read that a human starts getting into this kind of trouble if they drink more than 2.5 liters of water at one setting fairly quickly. They've made a hypotonic situation and their cells can start bursting. In a plant, that's what they want. They want hypotonic, hypo, bigger and round. And so that central vacuole gets big, puts turgid pressure on there, and suddenly we have turgid pressure. We have a plant that doesn't look wilted. Why does that work? The cell wall is really strong with cellulose in it, and that keeps it from bursting, which is what would happen in an animal cell. So turgid cell from a leaf with enough water, and then flaccid cells, so we could go even the other way. The poor guy's almost about dead. The other solution is hypertonic. I always like to think of my husband. He's very thin. He's also hyper, and so I think hyper gets smaller, thin. So if these are in a hypertonic solution, that means the higher concentration of water is inside the cell, lower concentration of solute. Out here, for example, there's more salt than water, and so the water is going to move out to balance, and we get shriveled cells. Bad for us. That's when we're dehydrated. If you go to the hospital, they have to actually give you an IV to try to hydrate, and they'll give you a hypotonic solution to try to bring those back up to isotonic. Up down here, poor plants, they really have problems. They go all the way to actually something called plasmolysis, where it pulls away from the inside of the cell wall and really shrivels all down. Doesn't come back from it very good. So I told you, a patient arrives at the hospital. She had the flu, severely dehydrated. What are they going to put on her? A hypotonic solution to make it swell up like a hippo so that they'll gain some water. So tell me, is this a hypotonic or hypertonic? Hypo makes them get bigger. Hyper makes them get smaller. Salting beef jerky as you try it. Hypertonic, making pickles with the solution. More salt on the outside, more sugar out there. Hypertonic. After eating potato chips, your lips are dry. That salt in there makes around your lips hypertonic. The misting sprays on vegetables at the grocery store. You walk along and they're spraying them. That makes them hypotonic. They absorb that extra water because it makes them bigger, remember? And then they seem nice and firm and wonderful, and you want to buy them. Putting sugar on sliced peaches to make them syrupy. Hypertonic, you're making that water come out of it. All right. I think you get the gist on that. Let's look at some osmosis problems a little bit. So we have solution in a beaker over here in the beginning, a 5% salt in the beaker, 10% in the tube. So what's going to happen? The water's going to move. The water's going to move into that tube to try to get the same ratio of solute, which is sugar, to water inside that tube as out. So we get more water going in there. Let's look at another one. This is like in the test, but different percentages. Let's say the beaker contains 10% protein, 12% salt, 10% glucose. The tube contains 5% protein, 6% salt, and 8% glucose. So the difference in here is this membrane allows salt, water, and glucose to move across, but protein cannot. So what's going to happen? All of those, salt, glucose, and water, are going to even out on both sides after 12 hours. But the protein can't move. So, which contains a higher solute content, the beaker or the tube at the end of your time? The protein remains higher in the beaker. It was 10%, so it's going to have So because everything else was able to move except the protein, we end up with a higher protein percentage in the beaker, so it is hypertonic to the solution that was in this tube. Let's do another one. All right, look at our things. They're both 5,2,10. This is 5,2,18. Once again, we're saying salt, water, and glucose can move. They're going to level out. So what do we have? Isotonic, because protein is going to be equal on both sides because it started out that way. And that's the one that's not going to move through there. All right. So we have active transport. This is the one that's going to take energy. This takes that protein with a specific kind of molecule it's going to pick up, move across. And it's going to take ATP to do it. So ATP is going to turn into ADP. That phosphate's going to pop off, energy is going to be released, and that guy's going to move across. Boom. Got it. Those are sometimes called pumps. A sodium-potassium pump is important in your body to maintain the ions and nerve impulse conduction. One of these pumps is actually broken in someone with cystic fibrosis, and that is what creates the buildup of mucus in the lungs, as a matter of fact. All right, watch that. So we can have bulk transport. Macromolecules are too large, and so we use vesicles to get them out, in and out. So let's talk about exo, means to exit. Cytosis is cell, so this is movement out of the cell. And so we have this vesicle full of stuff that we need. Perhaps it's a hormone that's been excreted and built by that cell. So it moves to the cell membrane, it merges with it, and then it opens it out so that little bit of whatever that we needed is released. Also, we take our waste out of the cell this way too. So when neurons release the neurotransmitters, it says in your brain, let's say, exocytosis is what's happening. Your neuron has this little bit of molecule that's going to cause some type of a reaction, goes out here, it's released, merges, release, causes synapse to occur, and a second impulse is then stimulated in this other part. Endocytosis, endo, to take into the cell. Endo means to take in, cytosis, cyto means cell. Phago, to eat the cell. So this is the sad end of a red blood corpuscle by a white blood cell, by a monocyte. So here are the differences. Pinocytosis is around a liquid. This is receptor mediated. It means that it's a specific chemical is looked for, and there's a little receptor there that makes sure the right one comes through. So endocytosis comes the same way. They're right here, it bubbles in, closes off, and now we got it. And here's some actual slide images of a receptor mediated. You'll see these little receptor guys right here, and those are the ones that look for a specific molecule. And so it's a receptor protein that picks up and concentrates that molecule in there. And that is it. Good luck.